Hello and good morning everyone. So today now we will be discussing on, we are continuing our concepts on chapter 4, that is the innate immune response. We have previously discussed about this innate immune response, the inflammation and elimination of the viruses. Then we have talked about this entry of the microorganism, barrier, physical barrier are breached, then get into the tissue where the innate immune cell, that is the macrophage, neutrophil, dendritic cell comes and takes the role. If the tissue are breached and gain into the blood, then there is the alternate complement pathway that activates, and by this way they will try to control the infection, they will try to eliminate the infection. If they are not able to do that, then they will take the help of the adaptive immunity, that is antibody dependent immunity, acquired immunity. Talking about the innate immune components, we have talked about this anatomical barrier like skin and then respiratory tract epithelium and GI tract epithelium and the gastric pH as well as respiratory epithelium cilia that was propelling out of all the microorganisms. Then we talked about the physiological barrier where temperature, pH, chemical interference were there, and then there was the innate cellular response about the phagocytic cell, that is the monocyte, macrophage, neutrophil and dendritic cell that takes place the role when there is breach of your skin that comes into the tissue and they goes and attack those microbial pathogens. Okay, so they recognize those organisms on the basis of this pathogen associated molecular pattern, that is called PAMP, or they can even clear the disease or damaged cell inside our own body, and that is recognized by damage associated molecular pattern. So we have talked about different things like anatomical barrier, physiological barrier, and if they are breached, then innate cellular response comes and plays role and try to control the infection. They do by recognizing this pathogen associated molecular patterns and DAMPs, that is damage associated molecular pattern. Moving forward, initially we have talked that innate immunity system can recognize only few patterns, but when the adaptive immune response comes, they can recognize more than 1 billion specific sequences on pathogen. So obviously adaptive immune system, that is the monoclonal antibody system, will be actually more specific to deal with the infection, but they comes only after 1 to 2 weeks of the infection that enter inside your body. So initially it's the immune system responsibility to clear the infection. If they are not able to do, then obviously then adaptive immune system will, that is B and T cell, will comes and play the role and clear all the infection and eliminate the infection. Moving forward, there is another term known as the inflammasomes, that is the part, that is actually the part of the innate immune system. This is part of innate immune system. It is expressed in the myeloid cell as a signaling system for detection of pathogen and stressor. So we know that we have talked about this lymphoid and myeloid cell, so you can remember all these things. So we have talked initially about the lymphoid and myeloid cell, lymphoid and myeloid origin. So that was there actually. So the lymphoid is T and B cell that you know on already, but coming out of the myeloid cell, we have the myeloid series, that is actually B, that is neutrophil, this macrophages, monocyte, dendritic cell, eosinophil, mast cell, basophil, which we have talked about this. If you see over here, this is the multipotent stem cell from where we are talking about this lymphoid originator, that is the lymphocytes, and there is the myeloid originator that is due to the interleukin 3 and GM-CSF, that is granulocyte monocyte coloring stimulating factor, that's reached to this myeloid origin. Myeloid origin we have granulocyte, that is neutrophil and macrophage, eosinophil, basophil, megakaryocyte and erythrocyte progenitor. These all are the myeloid stem cell, and this myeloid stem cell has this process that recognize this inflammasomes. So that is important actually. Okay, so let's move to the myeloid stem cell where inflammasomes, this is one of the way we deal with the things. Okay, so myeloid stem cell, so inflammasome. So inflammasome is an important part of the innate immune system. It is expressed in myeloid cell as a signaling system for detection of pathogens and in the stressor. So these are present in myeloid cell, which we have talked about, like macrophage, neutrophil, dendritic cell. They are present inside them, and how they are expressed, how they act, how they activated by the stressors and the pathogen signaling the system for detection of the pathogen and the stressors. So the myeloid cell, they are present in the myeloid cell, they detect the pathogen and stressor and then get activated. Activation of what happened when this get activated? Activation of this inflammasomes result in the production of interleukin, that is the chemokines, interleukin 1 beta and interleukin 18, which are the potent inflammatory cytokines. So that is important, actually. You have to understand this is inflammation of the inflammasome that result in the production of interleukin 1 beta and interleukin 18. That is important. They are present in the myeloid stem cell and they are the triggering factor is the detection of pathogen and the stressor. Okay, so moving forward, so this is your inflammasomes. So in inflammation, what happens if there is a stressor, that is the pathogen or stressor? You can see this is a plasma membrane inside. This is the myeloid cell. You can say think about the macrophage or neutrophil or something like a dendritic cell. Inside this, they have this NLRP3. This NLRP3 is a sensor. There is an narrator, and there is the caspase 1. So this is the NSP3 inflammasome. When there is the stressor or pathogen, then what happens? This NRP3 sensor and adapter get initially caspase 1 is in inactive form when they are bound by it, but when they get detached, then this caspase 1 get activated. And once, why this caspase 1 become activated? Because presence of this signal, that is the stressor or the pathogens that had been detected by this NRP3 sensor. So they had been the inactivated caspase 1 converted into the active one. And so once the active one has been formed, what they do, they will actually form this pro-interleukin 1b, pro-ligand to interleukin 1 beta into the interleukin 1 beta that is secreted outside and plays the role in the acute inflammation. So what is happening? We have to understand this caspase 1 is required for the activation of this pro-1 interleukin 1 beta into interleukin 1 beta, and that is how it will come. It will come by this inflammasome. When the stressor or pathogen will be detected this by NLRP3 sensor, they will activate the inactive caspase 1 and become the active, which will then convert this pro-1 interleukin 1 beta into the interleukin 1 beta, and then that will release outside and plays an inflammation role that will cause the inflammation. So hope you have understood about this inflammasome. It is a part of your innate immune system. They are present in the myeloid cell, that is neutrophil, macrophage, dendritic cell, and then they plays the role. So the release of the secretion of interleukin 1 beta, which is responsible for your inflammation. These are the cytokines that is released by the innate immune system. That is initially the pathogens or the stressor is detected by this NLRP sensor, and if they have detected, then they will convert the caspase 1 into the active one, and this caspase 1 active one will actually form this interleukin pro into the interleukin 1 beta, and then that will be released outside and cause the inflammation. So this is the way how innate immune system is dealing the infection. They have not gone to the adaptive immune system. They are trying by their own to eliminate. Initial what was the initial part? Initial was your anatomical barrier. They are trying to prevent the organism to enter into the body. There was another physiological barrier that is also preventing the organism or inhibiting the growth of the organism. Then there was the innate cellular response. That is once the pathogen has entered inside your cell, now what happened? This cell, that phagocytic cell, this neutrophil, macrophage, they comes and play into role and try to clear the infection. If they are also not able to do, then they are activating this inflammasome that is releasing the cytokines. This stressor and pathogen detected by this NLRP3 sensor will activate this caspase 1, which was initially present in our body in inactivate form. When they get activated, they convert this pro interleukin 1 beta into the interleukin 1 beta, which is secreted outside, and now the inflammation has begun by the innate immune system itself. So now moving forward, there are some receptors of innate immune system. You have to understood that this toll-like receptor, innate signals, this toll-like receptor was responsible for transcription of this pro-interleukin 1 beta. So what I'm trying to make you understand this toll-like receptor. There are few receptors present on this macrophages or say dendritic cell or neutrophil. This myeloid stem cell originated phagocytic cell. They have this receptor inside their incorporated on their cell, so recognize the pathogen outside present in. So if any pathogen enter inside your body, if they recognize those pathogen with the help of this receptor, and these are called the toll-like receptor. There are three types actually: toll-like receptor, nod-like receptor, and RIG-like receptor. So these are, some are present in the extracellularly, some are present in intracellular endosomal, some are present in intracellular cytosolic, and some are present in the cytoplasm. So these are responsible for recognizing different varieties of pathogens. They can recognize bacterial product, they can recognize viral product, they can recognize fungal product. So they depending upon the other structure. Actually, they have their capacity to detect. Like, let me discuss once again, make you understand. This toll-like receptor is present inside in the phagocytic cell, your phagocytic cell. These are the detector to detect the pathogens. So this toll-like receptor, like 1, 2, 6, 2, then toll-like receptor 4 and toll-like receptor 5. 1, 2, here is no 3. So these are, except 3, this 1, 2, 3, 1, 2, 3 is not, 4, 5, 6. These all are extracellular. 3 is intracellular. Then 6 up to here. 7, 8, 9 is up till toll-like receptor till 9 is over there. We have discussing here. Among them 1 to 6, except 3 is present extracellularly. 3, then 7, 8, 9 is present intracellularly. And what they can recognize, like toll-like receptor present on your macrophage, on your dendritic cell, on neutrophil, that can recognize your bacterial lipopeptides. Toll-like receptor 2 will detect bacterial peptidoglycan. Toll-like receptor 4 can detect bacterial portion lipopolysaccharide. Toll-like receptor 5 will detect the flagellin. So these are present on your side, on your immune cell, and bacterial lipopeptides will be detected. Bacterial peptidoglycan can be detected. Lipopolysaccharide can be detected by toll-like receptor 4. Flagellin can be detected by toll-like receptor 5. Then there is a viral portion, double-stranded RNA will be detected by this toll-like receptor 3. Then a single-stranded RNA will be detected by toll-like receptor 7 and 8. Then unmethylated CpG oligonucleotide will be detected by toll-like receptor 9. So these are your receptor present on your cell, your phagocytic cell, your immune cell, and these are the component of their foreign body, that is the bacteria, virus component that will be detected by this toll-like receptor, and then they will activate your cell and then try to clear the infection. So activation of transcription factor including NF-κ-beta, which result in the transcription of cytokine, addition molecule, and enzymes that are antimicrobial. So toll-like receptor are your receptor present on your cell, that you have to understand, and they can recognize respective portion of the foreign body, like bacteria and viruses mainly, you have to understand. Similarly, there is a nod-like receptor which is also called intracellular, that is cytosolic. So they are nod-like receptor 1 and 2, that is component of the bacterial PG, peptidoglycan, signals behind NF-κ result in the macrophage activation. Then is the NLRP3, that is the process of inflammation. We have discussed this microbial product and macromolecules from the damaged or dying cell, that is ATP, uric acid crystal, reactive oxygen species. So these are the stressor that will activate the inflammasome. When inflammasome is activated, this NLRP3 sensor plus adapter protein like pro-caspases will activate this caspase, which will cleavage the pro-interleukin 1 beta to generate interleukin 1 beta. So there will interleukin 1 beta will be generated outside and cause inflammation. Due to this is the dead and dying cell that is releasing this stressor. Okay, and then there is the RIG-like receptor that is present in the cytoplasm, and they are responsible for detecting the viral RNA and then interferon production and preventing or preventing the viral infection or controlling the viral infection. So these all are your part of the innate immune system. Present, this receptor are present on your cell, and they can detect the respective pathogenic microbials and then try to eliminate them by innate immune system. So there might be some mutation in your cell, that is mutation signal molecule affecting the toll-like receptor, then you will develop recurrent or bacterial infection, because you know this toll-like receptors will detect this bacterial polypeptide, and if you have this mutation over there, they will not able to detect this bacteria product, bacterial surfaces, and then this bacteria will enter inside your body, they will be undetected, and you will develop the recurrent infection. So toll-like receptor, recurrent and severe bacterial infection, pneumonia, gain of function of mutation in inflammasome, result in the gout, atherosclerosis, and type 2 diabetes. If there is nod-like receptor there is mutation, then they will develop inflammatory bowel disease. Nod-like receptor will be developing in inflammatory bowel disease. If you have interleukin 12 receptor deficiency or interferon gamma deficiency, then you will have this mycobacterium intracellular bacterial infection. So you can understand, if your defense system cell cannot detect the signal of the bacteria, then obviously you will develop that bacterial infection. Talking about the sum of the cell, actually, in the innate immune cell, there will be the myeloid cell that you have now understood. This monocyte, macrophage, and neutrophil, they are present in the bloodstream. Monocyte is in the bloodstream. They, when goes outside and in periphery, they becomes the macrophage. Macrophage in the different places has a different name. Then in the lung, it has the alveolar macrophage. In the blood, it is monocyte. In the brain, it is neuroglia. In the liver, it is Kupffer cell. So in the different places, it is a different name. In the tissue, it is usually called macrophage. The same thing in the blood is monocyte. Okay, so they have, this is CD14 positive. You have to remember, this is CD14. All these are the CD14 positive. They are phagocytic cell differentiate into the tissue macrophage. This macrophage is present, has a ruffle membrane, cytoplasm with the vacuoles and vesicle. They are CD14 positive, and they are actually causing phagocytosis and secretion of the cytokines. Okay. Now talking about the neutrophil, neutrophil is the multilobulated. We have discussed about this. This is the 3 to 5 nucleus is present. There is present in the bloodstream. They are about 18 to 78 on 100 per microliter, my multilobulated nucleus. So that is important. Okay, that is the multilobulated nucleus. Small light pink to purple granules, and they all are CD14 positive. What they do? They cause phagocytosis and activation of the bactericidal mechanism. So we know these are our phagocytic cell, that is monocyte, macrophage, neutrophil, dendritic cell. So cells of innate immunity, neutrophil, monocyte or macrophages. Okay, and then there is a dendritic cell, mast cell, natural killer cell. These all are the part of your innate immune system. Okay, so talking about this in a neutrophil, they are circulating phagocyte, short-lived, rapid response, not prolonged defense. This will provide you a very short period of defense. Then there is the monocyte or macrophage. Monocyte circulates in the blood, become macrophage in the tissue, provide a prolonged defense, produce cytokines and that initiate and regulate the inflammation. Phagocyte pathogens clear dead tissue and initiate the tissue repair. Macrophage will develop along one of two different pathways. So they have, macrophage has actually two functions. One, they start your phagocytosis, and they also repair the tissue and control the inflammation. So that is also their part. This is the role of the macrophage. Dendritic cell, they are present in the epithelial tissue. They have, all are CD14 positive, antigen capture, transport, presentation. They are antigen presenting cells. So they will capture the antigen, transport, present, and initiate the inflammation. The mast cell are present in the tissue, mucosa, and epithelial cell. They are small nucleus, cytoplasm packed with the large blue granules. They are release the granules containing histamine, and this histamine during the allergic response, and that causes the vasodilation and the drop in the fall in the blood pressure. This is the natural killer cell. This natural killer cell was not part of the myeloid cell. These are the part of the that originate from the lymphocyte, and they are not CD14 positive. They are CD16, and they are the CD56 positive. You have to remember this CD marker because they are the one of the way to recognize this particular cells. They are used in the immunohistochemistry and a lot of places. So you need to remember this. Okay, they kill a tumor killer or virus target cell. They kill the tumor cell or virus cell, or they can even kill by antibody coated, targeted, and secretes this interferon gamma. So they can kill the virus, they can kill the tumor, they can kill the cell that is antibody coated, and then secretion of the interferon gamma. So then